Hello everyone, good afternoon all. It is Gautam and I am taking botany here. And we have discussed in the last two sessions about the sexual reproduction in flowering plants. So we have completed up to male gametophyte, that is pollen grain. So that in the last class what we have discussed we will revise with a quick recap. So we know that on May 3, NEET exam completed, and from this chapter there are four questions they asked from this flowering plant, sexual reproduction in flowering plant. They asked four questions. So we'll try to answer if we get that completed quickly. So we'll solve some questions also. So we completed structure of microsporangium. Sporangium is nothing but what? It is a bag, it is a sac. So it is the bag or it is the sac which contains the sporogenous tissue, that is microsporangium. So there are four are there. We call this as tetrasporangiate and bilobed dithecus. Two theca are there or two sporangium is there. So this it is having four wall layers. The outer one is epidermis, and the next to that is endothecium. Next to that is middle layers. 2 to 3 middle layers are there. Next to that, what you are seeing, the dense cytoplasm, the innermost layer, that is tapetum. So tapetum is the nourishment giving tissue, so which gives nourishment to the developing pollen grains. The pollen grains which are present here. So for the developing pollen grains, the nourishment will be provided by these tapetal cells. So we know about these layers of the cells and the features of tapetum cells and how it is functionable and what is the characteristic of tapetum cell, how it looks and what is the ploidy of this. So that we have discussed in the last session. And apart from this, we also discussed the microsporogenesis also. So I will make a quick revision of this one, quick recap of last class. See, initially there will be sporogenous tissue. So sporogenous tissue will be there. So sporogenous tissue, it will be in diploid condition. Diploid condition is nothing but diploid. Two set of chromosomes will be there. So if you take cell of my body, if you observe under microscope, you will be seeing two set of chromosomes of all 23. Means 23 into 2, 46 chromosomes will be there. 23 pairs will be there. Like that, if you observe under microscope of this structure of TS of anther, so the microsporangium is there, no? The microsporangium, young anther. So the young anther contains, so young anther is containing what? Sporogenous tissue here. So the young anther, it is containing sporogenous tissue. Sporogenous tissue in the sense it is having diploid chromosome will be there in the sporogenous tissue. This is in a young anther. So this, it will be in a sporogenous tissue, it will be in a diploid state. So next what happens? This sporogenous tissue, it will get converted into the some of these, all these sporogenous tissue are there, no, which is diploid in number. The chromosome, the each cell of this sporogenous tissue or the we call it as sporogenous cell. They are homogeneous. These sporogenous cells are homogeneous. Homogeneous in the sense all looks similar, shape, size, and so this sporogenous tissue, the cell, it will be converting into microspore mother cell. So it will form microspore mother cell or pollen mother cell. So it can be called as pollen mother cell. Pollen mother cell because it gives birth to the pollen or it gives birth to the microspore there. That's why we are calling this as microspore mother cell, which is also is in 2n stage. 2n stage. Initially the cell size was this much. Think that the cell size was this much. One cell size it is of this much. So it increases in cell size so that it undergoes a division there, so that increasing it converting that sporogenous cell into microspore mother cell. This microspore mother cell, it will undergo meiotic division there. It will be undergoing meiotic division or meiosis to form microspores or microspore tetrad. To form microspore tetrad. And this microspore tetrad, it will be in a haploid condition. So which was in diploid condition, here it is haploid. Means after meiosis, 4 daughter cells are formed and the 4 daughter cells will be attached to each other like this. So it is microspores. So it is microspore here, which is wet in initial structure. The initially it will be in a wet state. So later after maturation, these microspore tetrad, it will be, so each one is a microspore there. This one is microspore. So this is microspore. Each microspore it will be turning into pollen grain there. So turning into pollen grain. So it is a pollen grain there. So it is pollen grain. The microspore will turn into pollen grain. And pollen grain is of two types we will be seeing based on the cells which are present in it. So one is, so if the pollen grain undergoes mitotic division, it forms two cells, so that two celled stage will be seen. Two celled stage of pollen grains is seen. So if it is undergoes another mitotic division, two mitotic division, so another mitotic division, it will form three celled stage. Why not 4, why 3? Because see, two celled stage is nothing but it forms two cells here and the lower cell is there. No, only that it will be undergoing mitotic division to form three cell stage. That is the microsporogenesis and microgametogenesis also seen. The male gametophyte is also discussed in the last session. That is, cell membrane is there, and next to that intine is there, next to that exine is there. So this exine is discontinuous and it is having, it is made up of sporopollenin and it is hard and it is the hard substances which cannot be dissolved by any organic enzymes or any acids also. So that is exine, which is made up of sporophyllin and it is discontinuous and irregular in shape. And the next to that is intine and it is continuous shape and it is made up of cellulose and pectin. We have discussed in the last session. And the plasma membrane, it is also made up of it's like a normal plasma membrane of all cells. So it's a plasma membrane. And inside of that we will be seeing two cells and one cell which is having bigger in shape, that we call this as vegetative cell, and which is smaller in shape and which is spindle in shape, that we call it as a generative cell. This is generative cell which is in spindle shaped structure like this, and the spindle nucleus will be there within that. So that is two cell stage. This is two cells are there within the male gametophyte. Male gametophyte is nothing but it is, they are talking about pollen grain here. They are talking about pollen grain. So this is generative cell. This generative cell, which is smaller in shape and smaller in size, this generative cell will undergo another division to form two male gametes there. These are the gametes. So you should know the difference between male gametes and male gametophyte. Male gametophyte is nothing but pollen grain. Here gametes is nothing but these two generative cell. When it divides like this, so it forms two gamete cells there. So when it divides like this, two cells are formed. Okay. So it is about the male gametophyte. So the male pollen grain is ready now. So the pollen grain is ready now. The male gametophyte is ready. And before that, we will be discussing about some pollen grain allergy. See, not all pollen grains are good. Not all pollen grains are allergic in nature. So if you visit the Nilgiri planted area, Nilgiri plantation region, or the some of the if you visit Bangalore also, you will get to know there are some seasons where the pink flowers are blooming and the yellow flowers are blooming. So you can see that all the trees will be blooming with the same color of flowers there. So there is some yellow shower that is golden shower plants are there, which will be seasonally like they will be flowering there. So this at the time they will be releasing the pollen grain in the air. So at the time the air will be carrying the pollen grains that we call it as airborne pollen grains. So when the pollen grains are moving in air, so that it may affects our body. So this pollen grain, it causes some allergy also, like it creates bronchial afflictions. Bronchial afflictions in the sense, see bronchi, you know that in human physiology we learned that. You already learned. So which is having bronchi, it is there here, trachea and this one. So this, it will get afflictions is nothing but it inflamed. It inflamed when the pollen grains enters in our lungs there. So bronchial afflictions is seen. Some they cause us asthma also, asthma and bronchitis is also same there. So so this is some of the allergies. So in foreign countries, even they will be signboards also used like pollen season ahead. So we need to cover the mouth, nose there, so that the pollen grains should not affect our nose, and it should not enter our lungs and the this trachea there. So if you take this plant, you might have seen. So it is everywhere, ubiquitous in nature. The NCRT line says the Parthenium, which came to India as imported wheat, along with the imported wheat variety. So that's so this is Parthenium or carrot grass. This is Parthenium plant, or it is also called as carrot grass. It is also called as carrot grass. Even these pollen grains are there, no? This plant pollen grains, it is allergic to. It is it is allergic to. So it is allergic, and it came to India along with the foreign imported wheat variety. So it came to India, so along with the wheat imported variety there. So wheat imported variety, it will be it comes to India. So that is Parthenium species or carrot grass. So that it is everywhere now. So that is also causing some allergies there. So this pollen grain, it will be like even in our India also, you can go and just check uh, like where the plantation is done. You can, if you just go without covering your nose, you immediately you may sneeze, or you will get some infections there due to the pollen grain if it enters our body. So will our body will be creating some defense mechanism there, so that due to that will be some allergic reactions is in our body. So is that the pollen grains only has the demerits or losses, or which are harmful? Means, so uh, I don't think that the pollen grains only are having harmful effects on our human. So they are some pollen grains are which are useful also, which are beneficial in nature. So that you can see here. So this is bee pollen. So some of the company which is like bee pollen grains. So the pollen grains benefits are, it is pollen grains are rich in nutrients and vitamins. So these are pollen grains are rich in vitamins and nutrients. So due to that, these pollen grains can be consumed by the athletes. In athletes, they will consume this pollen grains in the to enhance the performance. Not only athletes, even in the race horse also. Like see, you can see the pollen X, which is a bee pollen or pollen feed for the horses. It is seen in pollen feed for the horses. So where we will be feeding to the horses for even in athletes, I mean even in the race horses also. So this it will enhance the performance here. So this pollen grain they will be enhancing the performance in case of athletes and race horses. So this pollen grain can be used in the form of supplements there, so that even they can be taken, these pollen grains they can be taken in the form of tablets or syrups. So these are like supplements for the, so it can be taken as supplements to which enhance the performance in case of athletes and even in the race horses. So that you can see that in abroad and all they will use a lot of this one to feed the race horses there to enhance the performance. So these are the pollen grain benefits there. So they are rich in vitamins and nutrients, so that they can be used for in case of race horses and athletes there. So in the form of tablets and syrups. You can see in the NCRT lines they have given the some tablets images and all. So next is the pollen viability. Pollen viability in the sense here viable word is used. Viability is nothing but survivability. Survivability is nothing but see the pollen grain is matured and it bursts. The anther bursts. When the anther bursts, the pollen grains will release in the air. So after bursting, after releasing of pollen grains, how much duration they will be surviving and they will be showing the germination. Germination is nothing but germinating the pollen tube here. So you can see here. So this is the pollen grain and there is a germination of pollen tube here. This is the pollen tube. So pollen tube it is. So after this is germination of pollen tube after the releasing of the pollen grain. So the in different plants the pollen viability is differ. So the pollen viability here it differs in different plants. So in case of rice and wheat, so in case of rice and wheat, so the pollen viability lasts around 30 minutes. So only of 30 minutes it will be the pollen viability will be survived for pollen grain will be survived for 30 minutes. After that the pollen germination will be taking place in case of rice and wheat. But in case of some species or some family there, so RLS you need to remember. RLS is nothing but Rosaceae and Leguminosae. So these are the families and Solanaceae. So these three, Rosaceae, Leguminosae and the Solanaceae, so there they will maintain the pollen viability for several months. So the pollen viable, it will be viable for the several months, like around 3 to 6 months. So that is Rosaceae, Leguminosae and Solanaceae. There the pollen grain can be viable or survivable for several months, 3 to 6 months it will be survived. In case of these Rosaceae is nothing but you can take example of rose. Leguminaceae. Leguminaceae is nothing but like green gram. You can take pea, peanut. That is Leguminaceae. And Solanaceae. So Solanaceae we have learnt in the last class 11. That is tomato, potato, brinjal. So there the viability lasts for 3 to 6 months. And next is pollen storage. Pollen storage is nothing but the pollen grains can be stored in the form of in the pollen banks. So these pollen grains can be stored also. So what are the conditions which are required to store the pollen grains is that is it requires minus 196 degrees Celsius in the liquid nitrogen. The pollen grains can be stored in minus 196 degrees Celsius. So this process, the storing of pollen grains or the storing of pollen grains or plant tissue in this minus 196 degrees Celsius in liquid nitrogen, that we call this as cryopreservation. That we are calling it as cryopreservation. Cryopreservation is nothing but storing the pollen grains or in minus 196 degrees Celsius. Even if you check the PYQ, they asked this question. Previous year Neet you can see this question and the pollen storage or pollen banking. So why the pollen grain storing is required or pollen grain can be stored and preserved because so it is used in crop improvement programs. Crop improvement programs in the sense if there is some plant is there which is having unique character. So I cannot see that character in some other plant. So in that case, what I will do is, I will preserve the pollen grain of that particular species. So how to preserve it? So we will preserve in this condition. So that process we call this as cryopreservation. The pollen grain which will be stored in the form of banks, like pollen banks are there. So these pollen banks, there it will be used for crop improvement program. And not only that, even to preserve the plant species, for conserving the plant species is also seen there. So this is for conserving the plant species also. So where the pollen banking is done, so preserving the pollen grains under liquid nitrogen conditions of this much eventually. Pollen grain preservation. So it is about the pollen viability and pollen storage. So the things what we have learned so far is we have discussed in the last session about the pollen grain and how the pollen grain is there and what is the structure of it, what is the function of each layer there and how it will germinate. So through the germ pore it will germinate. So when the through the germ pore when the pollen tube formation takes place, so this is vegetative cell. So vegetative cell will guide the generative cell. It will guide the generative cell to reach out the female gametophyte there. So here you can see tube nucleus is nothing but the vegetative cell. It is guiding the generative cell here, generative nucleus. So it is in two cell stage. So when the pollen grain is released, there you will be seeing two types, that is two cell stage and three cell stage. So this generative cell will undergo again division to form two cells there. So that is male gamete. So this generative cell shown the division there. This it forms mitotic division will be taking place in generative nucleus to form three cell stage. Two male gametes are ready here. So this is about the pollen grain and the pollen germination and the pollen viability. Viability is nothing but the surviving of the pollen grain before it after releasing from the anther and before the germination, that is pollen viability we call it as. And the pollen viability is different in different species. It is varies. So in case of rice and wheat, it is it lasts for around 30 minutes. Whereas in case of Rosaceae, Leguminaceae, and Solanaceae, it lasts for several months or 3 to 6 months. That is about the pollen viability. So next we will be discussing about the pistil here. So pistil is nothing but it is the female reproductive structure. So even we can call this as carpel also. This is carpel. So what we are going to discuss in today's section, today's session is that is female reproductive structure. Female reproductive structure, that is pistil or carpel. So what the pistil is composed of? So if you take drawing the pistil here, it is the pistil here or the carpel, and the parts here are the landing surface is there. No, here the pollen grain will come and land. The pollen grain will come and land on this surface. The landing surface of pollen landing surface, we are calling this as stigma. It is stigma, and the tube-like structure, the slender tube-like structure, this one we call this as style. And the swollen base, this one we are calling this as ovary. So together it forms a pistil there or carpel. Pistil also same, carpel is also same, female reproductive structure, that is this also same. Or the gynoecium also we can call this as gynoecium. So that is gynoecium also we can call. Here stigma is there, that is where the landing, and through that the germination of pollen tube it will be taking place, that is style, and it reaches the ovary. So here ovary is there. So within the ovary, ovule will be there. So within the ovule, within the ovary, ovule will be there. And before that, it will be having a placenta here. The placenta is there. Placenta is the nourishment tissue to the developing ovule there. That is the placenta. And to that you will be seeing the ovule is connected. The ovule is connected here. So the ovule will be connected to this placenta. Here what you are seeing is a cushion like structure, that is nourishment tissue, that is placenta is there, and there is a ovarian cavity. So within the ovary you will be seeing ovarian cavity, or we can call this as locule. We can call this as locule also. And within that the structure what you are seeing, that is ovule, which is attached to the placenta. So this is this is about the structure of pistil here. The pistil contains three structure mainly, that is stigma, style, and ovary. And within the ovary, the cavity, the empty space, the ovarian cavity or locule we call it as. And within that you will be seeing a ovule which is attached to the cushion-like structure, that is placenta. So it is about the pistil. And let us come to the some terms which we will be seeing in NCERT lines, that is. So the pistil or carpel, if the carpel, if it is present only one, we call that kind of plant or that kind of flower as monocarpellary. That is, we call this as monocarpellary. Monocarpellary in the sense which is having only one carpel. If only it contains only one carpel, it is monocarpellary. If it is containing two carpels, it is bicarpellary. If it contains two carpels, it is bicarpellary. If it is contains more than two carpels, then it is multicarpellary. So then in bicarpellary or more than one carpel is present, in that condition or in that cases, we will be seeing two types of flowers there. So one is apocarpus. So you already learned in morphology of flowering plants, 11th grade. So apocarpus condition, and another one is syncarpus. You need to tell me that what is that apocarpus and syncarpus. Here apo word is there. No? What is the meaning of this apo word? So here bicarpellary is nothing but more than one carpel is there, and two we will be seeing apocarpus and syncarpus condition. So this apocarpus is nothing but a free carpel. Free carpel is there. Syncarpus is nothing but fused carpels. If the carpels are fused in apocarpus condition, so there will be seeing of two carpels in a flower. Same flower, we will be seeing two carpels like this. So in case of syncarpus conditions, what happens? We will be seeing two carpels, but they are fused. The base is fused here. The base is fused. The base is fused. That is syncarpus. Fused condition, we call this as syncarpus condition in case of that one. And some examples is also given in your book. So that is right side of the corner side, you will be getting images there. That is syncarpus ovary. Syncarpus means fused. Apocarpus means free. Only one carpel is there. That even in this case also it is, you can see, and one carpel also you can see there. So in case of syncarpus condition, so where the carpels are free here, so the syncarpus conditions it is fused, whereas in apocarpus condition it is free. And the examples which are given, so the syncarpus is papavera, papavera somniferum is given, that example is given. Apocarpus are free carpels which are present in flowers, that example is given as Michaelia. So the examples is also important in case of this one. The examples are so examples for apocarpus flower, that is Michaelia, Michaelia champaka is given, and the example for fused carpels or syncarpus condition, that example is papavera somniferum. Papavera. So I will show the images, you will get to know. This is the syncarpus ovary. Syn means fused. See, you can see here stigma are many stigma are there. See, this is fruit. I am not showing this one. So you can first you see observe the flower, then you can go for fruit, and this is a flower here. So you can see here many stigma are there. Many stigma are there. Here many stigma are there. Many stigma are there. Many stigma, but means they are fused here. Ovary is fused. So it is ovary is fused here. Fused condition is nothing but syncarpus ovary. And the example for that, it is papavera. And after the fruit formation, what happens is, fruit is one only, single fruit, but it is having many stigmatic surface there, many stigma, many many stigma and style will be there, and the fused ovary is seen, that is syncarpus ovary. Example for this is papavera, papavera or papavera somniferum. And this is apocarpus condition. Apocarpus is nothing but you can see the image here. This is the NCERT image where you can see it is one carpel and it is another carpel. So they are free, they are not fused. You can see there. This is another carpel. So like that, the carpels are free here. They are not fused at one end. So this is the real image of Michaelia. So you can see this is one carpel and this is another carpel here. This is another carpel here. So the carpels are free here. The carpels which are free, we call this as this carpels which are free here. This is apocarpus condition. Apocarpus, which the carpels are free. And example for this is, example for this is Michaelia. This is Michaelia here. Michaelia is the plant name. This plant name we call this Michaelia. Here it is syncarpus flower with carpels are fused. Its carpels are fused, and example for this is Papavera somniferum or papaya. And going back to here, so that is we have discussed this thing. And coming to the ovule, see in this year, so one ovule is there. In one carpel, if it is, or in one flower, only one ovule is there. That is, it will be carrying only one seed. Within the fruit, we will be able to see one seed in that. For example, you will get to know many. That is monocarpellary condition, where only one ovule will be there. So in such cases, examples like mango you can take. If you when you are eating mango, only one seed will be there within the fruit there. Or if you take example of watermelon, so many seeds are there within that. So that is the multicarpellary condition. The examples is also important here. So if single ovule is present, single ovule, only one seed will be there. Only one seed is there. So example for that, mango. Mango, you will get only one seed there. So there is many seeds or many ovules is there name. Many seeds are found. Example like watermelon. Watermelon is there, and even orchids also is given. Orchids, in case of orchids also you will be seeing many ovules or many seeds. So this is, if it is having single ovules, only one seed is seen in case of plant. So I will show the NCRT lines also there. You can check. You just take the NCRT lines also. So in NCRT lines it is given below that diagram. You will be able to see the examples which is given there also. You can check. So here you need to remember the orchids mainly, because this is the you might have not seen orchid fruit there where it is having many ovules or many seeds there. So orchids is one example where within a flower we will be seeing many ovules. If only one seed examples you will get many, but uh, even in watermelon also we can see that many seeds. Easily understandable that many ovules are there, but orchids is the only example where we have not seen the. So here you can see. So these are the diagrams which are given. This is the hibiscus plant. where the stigma are many are there, then the ovary is here. So it is syncarpus and this one is there. So in the below that line you will be seeing placenta, the megasporangia, ovule. The number of ovules in an ovary may be uh, single, like in case of wheat or in case of paddy and mango, that the ovary will be only one ovary. So ovule will be the number of ovule in ovary may be only one in case of wheat and paddy and mango. In case of papaya, watermelon and orchid, so the many seeds will be there. You can see that. It means many seeds is nothing but many ovules. So far, what we have discussed is given here. You can check that later. NCERT line by line, you need to go. So here, so in today's class, what we have seen is that is 15 or the megasporangium. We are going to discuss that. In pistil, that is, we have seen that monocarpellary. If only one carpel is there, if more than one carpel or more than one pistil, that is multicarpellary. So where more than one pistil may be fused together, that is syncarpus, if they are fused together. And if they are free, that we are calling it as apocarpus. And the stigma style ovary, the pistil has three parts, that is stigma, style, ovary. And the stigma serves as a landing platform for the pollen grain to land on that. And the style is elongated slender part beneath the stigma, that is, we know that. So the swollen or bulged part we call this as ovary, and inside the ovary we will be seeing ovarian cavity or locule, and the locule contains placenta which is located in the ovarian cavity, which is a cushion-like structure, and which produces upon this placenta we can able to see the arising from the placenta we can see the ovule, or is nothing but megasporangia there. So we have discussed this one so far. So now coming to the megasporangium. So here megasporangium is nothing but it is the ovule there. So the megasporangium I am drawing here, like invertedly I am drawing. So it is the placenta. So placenta is there. Upon this placenta, from the placenta, there will be elongated structure so that the ovule is seen there. The ovule is seen there. This is the ovule which is attached to the cell. And inside of this, we will be seeing embryo sac. So this is the complete structure of ovule. Now let us label the parts of it. The parts of this one is placenta, we know that, placenta. And the connection from where the placenta, cushion-like structure, the placenta is there, the ovule is attached to that one. The ovule is attached to the, by the stalk, with the help of stalk there. The stalk of the ovule, we call this as funicle. Funicle is nothing but it is the stalk of the ovule. Funicle is nothing but it is the stalk of the ovule. And this is the funicle, and this is the junction. This is the main ovule here. See here. This is the funicle, the stalk of the ovule. And this part, what you are seeing, this part, what you are seeing, it is the ovule. It is the ovule. So this part, what you are seeing, this is the ovule here. This part is the ovule, and this part of the ovule, it is attached to this funicle there, and the junction where ovule and funicle is connecting, the junction where funicle and the main ovule is connecting, that the junction we call this as, this junction we call this as hilum. This is hilum. Hilum is nothing but the junction between the funicle and the ovule here. So it is the junction between funicle and ovule. That that junction we are calling it as hilum. That junction is nothing but hilum there. That part is this one. So this what you are seeing. This part is hilum. And next to that, what you are seeing here, this is the there are two layers you can see. This is the main layer, this is the ovule, and it is having two layers, one two. So this outer one is there. Now outer one we call this as outer integument. Integument is nothing but wall. So outer wall or outer layer we are calling this as outer integument. And next to that, the integument which you are seeing, next to that, what you are seeing, that is inner integument. The layer next to that outer integument or the wall layer, we call this as inner integument. And next to that inner integument, the what you are observing here, next to that inner integument, this layer is there, no? So which is this one. So this layer inner to the inner integument, the layer cells, the cells, a group of layer of cells are there, that we call this as nucellus. Nucellus is nothing but from where, from the nucellus cell, the further development of male female gametophyte will be taking place. Female gametophyte will be taking part from cell of this nucellus. So this nucellus, it is there. This nucellus has many cells here. So you can see it will be having cells here. Tissue will be there. So it is the nucellus tissue or the nucellus layer. So one of the nucellus cell here, from one of the nucellus cell, I am taking, let's consider this one. One of the nucellus cell, it will take initiative and it will act as megaspore mother cell. So it will act as megaspore mother cell. So then the megaspore mother cell undergoes meiotic division to form megaspore then. So from megaspore, embryo sac is formed. So this is done with the help of nucellus cells there. So the part of ovule, this kind of arrangement of this ovule is there, no? See, this is placenta, and the placenta, exactly to that placenta, this is there, the opening of this one. So the opening of this place, the opening of this ovule is there, no? This opening of this ovule, we call this as micropyle. This opening of this ovule, we call this as micropyle. Micropyle is nothing but the pore or the opening, small space which is kept opening for this one. The micro means small, pile is nothing but pore. Micropore, that is micropyle. And the swollen base, the base is bit swollen here. The swollen base, we call this as chalaza or chalazal end also you can call this. Chalazal end is nothing but the swollen. Chalaza word indicates swollen here. Swollen base. That is the swollen base of this ovule is chalaza, and opposite of that it is having a micropyle, that is the opening of this, or the gate, that is the opening entry of this ovule, that is micropyle. So this opening we call this as a micropyle. Micropyle is nothing but the micropore from where the pollen, usually pollen tube will be entering the this female reproductive structure there, that is in the ovary or ovule. So this megasporangium, this is the whole structure is megasporangium, or it is also called as ovule. And this type of ovule is there, no? This type of ovule, we call this as anatropous ovule. This type of ovule, we call this as anatropous ovule. Anatropous ovule in the sense, see, placenta is there, and it is the ovule is inverted and the micropyle is towards this one, this placenta. So there are different types of ovules are there, like orthotropous ovules or synotropous ovules. So like that, it is anatropous ovule. In case of orthotropous ovule, what happens? So it is the ovule. So these are the integument, and it is connected to this placenta like this. So the placenta is here, and it is exactly in the straight line, the micropyle will be there opposite to that one. So this is the orthotropous ovule. So this is the ovule which is given in NCERT, or we need to follow this one. That kind of ovule, we call this as anatropous ovule. So there are 5 types of ovules are there. No need to worry about all those. Only you need to remember this word. This word, that is anatropous ovule, is there for our exam. So this you can see the NCERT diagram. Phylum is there. Is the junction between the ovule and the funicle. Funicle is there. That is the start of the ovary. And micropylar pole. This is micropylar end. And this is the outer integument. And this is inner integument. And next to that, the layer which you are seeing is nucellus. And next to that, what you are seeing is embryosac. And next to that, the swollen base, what you are seeing is cella sulfate. And this is the microscopic diagram. Means when you observe under microscope, you will get this kind of ovule structure. So it is the anatropous ovule only, we can see here. So like this, it is connected, and this is embryosac, and these are integuments here. So this is a microscopic structure of ovule, anatropous ovule. So in the next session, we will be seeing megasporogenesis, how the megaspore is formed from megaspore mother cell. So in this session, what we have seen, one is pollen allergy, and next we have seen pollen benefits, pollen grain benefits, and pollen viability and pollen storage, and pistil, and next to that the types, the parts of the pistil we have seen, and next to that parts of the pistil we have seen different types of syncarpus and apocarpus ovule, and next to that we have seen the megasporangium, that is anatropous ovule. So far what we have discussed. So regarding this, the questions we will be discussing in the next session, and continuation of this, we will be seeing the megasporogenesis, how the megaspore will be forming, that we will be seeing in the next session. So till that, if you have any doubts or if you have any questions, you can leave a comment in this video. So I will be discussing that question in the next class. If you are not watching this complete video, you can watch it in 1.5x or 2x, so that you can cover the whole video in less number of minutes. So till that, goodbye. Thank you.